Microbiome workflows are fraught with biases and inconsistencies that can skew results. In fact, the same stool sample can yield contradictory profiles when processed by different labs. For example, one analysis found Bacteroidetes, gram-negative bacteria, to be dominant, while another found Firmicutes, gram-positive bacteria, dominating in the same sample. The likely cause was lysis bias. Suboptimal cell lysis in one workflow left many tough gram-positive bacteria underrepresented. This lack of reproducibility highlights why quality control is essential in microbiome research. How do we tackle bias? The solution is to use microbiome standards and controls as reference points. The principle is simple. Run a well-characterized mock community of known composition through your workflow to see how the outcome compares to what it should be. These standards are used in parallel with real samples as quality controls, so any deviation in the standard's results flags a bias in the process. In other words, a standard provides a ground truth, allowing you to calibrate your methods and troubleshoot issues by comparing expected versus observed results. There are multiple types of controls available, each designed to detect biases in different parts of the workflow. Two of the most important are whole cell standards and DNA standards, which we'll explore next. Whole cell mock community standards contain intact microbes and are run through the entire workflow, from cell lysis and DNA extraction to sequencing and analysis. They act as a positive control for the full process, revealing any biases introduced in the upstream steps, like inefficient lysis. Zymo's whole cell standard, for instance, mimics a complex community of known composition with microbes of varying cell wall toughness, including easy-to-lyse gram-negatives and tough-to-lyse gram-positives. Because all species are present in equal known abundances, you can directly compare your sequencing results to the theoretical profile. If certain groups drop out or inflate, you've discovered a bias. For example, if the gram-negative bacteria in the standard appear overrepresented, while the gram-positive bacteria are underrepresented compared to their true equal amounts, it's a red flag that your lysis method isn't fully breaking open the tougher cells. In this way, a whole cell standard tests your DNA extraction efficiency and lets you optimize upstream steps to ensure even the hardiest organisms are being detected. In contrast, a DNA mock community standard is made of purified genomic DNA from a known set of organisms. No whole cells, and thus no lysis required. You introduce this standard after the extraction step, directly into library prep, so it specifically tests the downstream processes like PCR amplification, library preparation, sequencing, and bioinformatics. By removing the cell breaking step, a DNA standard eliminates any extraction variables. This makes it ideal for pinpointing biases such as PCR amplification bias, for example GC content bias, or sequencing preparation errors. Researchers use DNA standards to optimize library prep conditions and validate their bioinformatic pipelines. If you sequence a DNA standard and the results don't match the expected community profile, you know the error lies in those downstream stages, since all the DNA was there in the correct proportions from the start. In short, the DNA standard serves as a control for the latter half of the workflow, ensuring that once DNA is free and available, your process amplifies and sequences it without distortion. Using both types of standards together gives you a comprehensive view of your workflow's performance. Here's how. Run the whole cell standard through the entire process, and run the DNA standard starting from library prep. By comparing the results, you can pinpoint where any bias is introduced. If the whole cell standard shows a skewed community, say loss of gram-positives, but the DNA standard result looks correct, that discrepancy reveals the bias occurred during the lysis extraction stage. If instead both standards show a similar deviation from expected, the problem must lie in the downstream step common to both. For example, a PCR bias, or a bioinformatic error affecting both runs. This two-pronged approach essentially isolates the variables. You can quantify how much inaccuracy comes from extraction versus how much comes from later steps. In practice, Zymo research recommends first optimizing the downstream workflow with a DNA standard to eliminate library prep and analysis biases, then running a whole cell standard to see if any additional bias appears during the extraction phase. By sequentially deploying the DNA and whole cell controls, you ensure that each part of the pipeline is finely tuned and any lurking biases are exposed and addressed. The ultimate benefit of these QC controls is confidence in your data. By routinely including standards in your microbiome workflow, ideally in every run, you can immediately detect when something is off. For instance, if your whole cell standard suddenly yields much less DNA than expected, or the community profile shifts, you'll catch it right away. This allows you to troubleshoot before processing valuable study samples. Consistently using these controls means the only surprises in your data are biological, not technical. Labs around the world have begun adopting this practice. In fact, hundreds of labs globally now use Zymo's microbiome standards to improve data quality. This growing culture of quality control is pushing the microbiome field towards much higher reproducibility and reliability. By eliminating hidden biases, we can finally compare results across studies and sites with confidence. In summary, mastering microbiome QC with whole cell and DNA standards ensures that your microbiome insights reflect reality, not workflow artifacts. With bias under control, you can trust your findings and focus on the science, delivering real, actionable insights from your microbiome data.